I want to introduce Kiersten Ritchie. Kiersten is a PhD candidate in the bee program in the WSU Entomology Department. Her research focuses on honeybee reproduction and evolution. She attended the University of Washington and received her Bachelor of Science degree in biochemistry. After graduating, Kiersten worked at the Hanford Nuclear Reservation before coming to WSU. She hopes to finish her degree this spring. All right, so Kiersten, we're looking forward to hearing about queen rearing. If you could take it away, I would be so happy. Thank you.
Hey, thank you for that introduction. Again, my name is Kiersten Ritchie. I'm a PhD candidate in the Bee Lab, and I have quite a bit of work experience with queens, mainly just in the time that I've been at WSU for the past 5 or 6 years. But due to the nature of my research, I've handled and done a lot of queen insemination. So hopefully I can give some good information on queen rearing, and I'll be going over kind of surface level information on queen development and some methods for queen rearing. So please, if at the end of this presentation you have questions, or if there's anything in this presentation that you wanted me to touch on that I didn't, please let me know. I'm always looking to improve these presentations to give beekeepers the best information possible that they are asking for. Um, so I guess with that, I will begin my presentation on queen rearing in the Western honeybee Apis mellifera. Oh, gotta start okay. So these are some of the resources that I've used for this presentation. There's a lot of great reading out there, physical book copies or on the internet. My specialty for queen rearing is in grafting, but I will talk briefly about some graft-free queen rearing methods, and this article by Morris Ostrovsky goes into great detail about that. So if you want any further reading, also some great resources for queen rearing and kind of creating your own breeding program can be found through the WSU Bee website or from Sue Kobe's website, and some great YouTube videos that our lab has put out. So to get right off the bat, within a hive, there are three castes of bees. You have your workers, your lovely queens, and your drones, which are the males of the colony. Workers and queens are both female, and the drones are the males, like I just mentioned. The worker is a female bee that hatches from a fertilized egg, and a strong colony will have tens of thousands of worker bees. The queen is also a bee that hatched from a fertilized egg, so genetically, a worker and a queen are identical, but as you can see, even just from this photo, they differ greatly in their physiology and morphology. There can only be one queen per colony at a time. The drones, on the other hand, hatch from unfertilized eggs. They don't do a whole lot for the colony, no defense or collecting food and resources. They are a bit of freeloaders within the colony, but very important nonetheless, because they are the ones that produce sperm that will eventually fertilize eggs from queens from other colonies. And their one goal in life is to store energy to make sperm and then fly out to mate. So a healthy colony should contain a limited number of drones, but up to a few hundred if it's really nice and robust and healthy. A good strong colony will be able to produce hundreds of drones to then proliferate the genetics from that queen. So honeybees have a unique genetic system. referred to as haplodiploidy. And this refers to the way that sperm producing male bees originate versus egg producing queens and the infertile female workers. Oh, goodness. Um, so just like in mammals, there are different pairs of chromosomes for males and females, but in honeybees, these chromosomes have, instead of multiple chromosomes for both males and females, the sex of a honeybee is determined on a single chromosome based on a single allele. So um, the queens and females are diploid, so they have two copies of this sex allele, whereas drones that are haploid, they only have one copy. So a queen produces eggs that have one set of these, one set of genes and one set of these sex alleles. And the drones will produce sperm that again have one single copy of his genetic makeup. And when the haploid eggs and haploid sperm come together, they create diploid progeny. So that the diploid progeny will either become workers or queens, and any eggs that are not fertilized by those sperm will become drones. In rare cases, when a worker becomes a laying worker, so in instances where a colony becomes queenless, the workers can lay eggs, but none of those eggs will be fertilized, and that's what happens when you have a drone layer in a colony. Is you typically a worker is laying eggs that only develop into drones. Mhm. So, regardless of the sex of the bees or their status as a queen or worker, all honeybees start out as an egg, whether it's fertilized or not. So honeybees have this life cycle that is holometabolous, and an easy way to remember that is they have a whole metamorphosis. So they go from through all of the life stages of an insect. So they start off as an egg, they move to a larva, and then several stages of pupa before emerging as an adult. All larvae, no matter what sex or life stage development those females are destined to, they're all fed royal jelly for the first 3 days after hatching, and then they are fed different diets to differentiate their development, especially if they're female. So, uh, even after all of the differences of haploid unfertilized eggs developing into drones and diploid eggs developing into females and being fed all of these different diets, um, each of the different castes take a different amount of time to develop. So queens have the shortest development time from an egg to emergence as an adult. It takes about 16 days. Workers from egg to adult is 21, and drones take even longer. They take about 24 days to emerge as an adult after being laid as an egg. So here you can see that for the first 3 days after being laid as an egg, this queen development chart shows that uh, all of the laid eggs will remain as eggs for 1 to 3 days before they hatch and spend about a week as larvae. And you can see the different stages of development from this little tiny grain of rice-sized larva all the way through this big girl up here before they develop into pupa and eventually emerge as an adult. And then even after the queen emerges as an adult, it takes further time to develop and mature before she will go on mating flights or be ready to be inseminated if anyone is wanting to artificially inseminate their queens. So for workers, it's pretty similar. You see that the first few days they're spent as an egg and then as larvae once they've hatched out of that egg after 3 days. But for each life stage after an egg, workers spend a few extra days in each life cycle development. So a few more days as larva and a few more days as pupa before they emerge as an adult on the 21st day. But as you can see from this image on the right, most cells will be capped on day 9 with some exceptions. Usually the rule is about 8 to 9 days is when the cells will be capped for queens and workers. And then with drones, as they take longer to develop, it'll take longer for their cells to be capped as well. And then further, just like as queens take more time to develop and mature after they emerge, it's similar with workers. So workers do all of the work in the colony. Like we said, drones are kind of freeloaders. Queens are there to produce eggs and new workers to beef up the colony throughout the season. So workers go through even further development, classified as age polytheism. So at each stage of development, these workers will be performing different tasks within and even outside of the colony. And a good rule of thumb to determine, what stage of development or how old these workers are is the rule of 3 with, there's always exceptions to these rules, but as a good rule of thumb, the rule of 3. So there are 3 brood stages from egg to larva to pupa, and then after that, they're adults, they're no longer part of the brood. So 3 days as an egg, they'll spend about 3 weeks as hive bees and then 3 weeks as field bees before they die. And that's typically for the summer season bees. Winter bees last a lot longer. So the first stage after a worker emerges is the hive, or what a lot of people refer to as nurse bees. These are the very young adults that have just emerged, and they will perform all of the cleaning within a hive. They will feed the young larvae with food from the different glands in their head, the hypopharyngeal and mandibular glands, depending on the type of food they're feeding. They will be ripening the honey, working with wax that they secrete from wax glands to shape and construct the comb. And then they'll also participate in guarding the hive and going on orientation flights before they become field bees, which is the next stage in their development. And these field bees are the bees that you see outside the hive, collecting nectar and pollen, as well as water to regulate the hive temperature, and propolis to help seal off the hive or the natural cavity that they may be residing in, as well as help fight diseases. So those nurse bees are going to be feeding developing larvae a mixture of different foods. But for the first 3 days of larval development, nurse bees will be feeding those larvae royal jelly. And this is a mixture of water, proteins, amino acids, fatty acids, and sugar that the workers will secrete from their mandibular and hypopharyngeal glands. These are located in the head of the nurse bee. And this is really good for development. It has low levels of toxins, like pesticides or certain plant chemicals that could be damaging or harmful for larval development. So again, like I said, all of the bees are fed royal jelly for the first 3 days after hatching. Um, but then after that, depending on what caste of bee they are destined for, mostly the female bees, um, workers will be fed worker jelly, which is a mix of adult food, pollen, honey, as well as more glandular secretions. But larvae that are destined to become queens are fed royal jelly for their whole life. So it's not completely understood how this change in diet completely changes the workers and queens physiology. Um, but the current idea is that there is a certain plant produced chemical in either pollen or nectar that are in worker jelly that essentially chemically castrates the worker. So it will um, slow down or completely reduce or stop the development of worker ovaries so that they are essentially infertile. This also prevents the cascade of ovarian hormones that would lead to the elongation of the abdomen, like you see in queens, as well as other queen-like traits like the production of queen pheromone. Mhm. So all of that to say that all of these stages of development and the production of royal jelly makes it easy for good, strong colonies to rear new queens for themselves if um, a queen is unexpectedly dies, or if she the hive swarms and the bees left behind need to rear a new queen. So here you can see there's uh, differences in the type of queen cells the bees will make depending on the status of the colony. So on the left, you have these nice, beautiful queen cells that are on the bottom of the comb, and this usually occurs when there's lots of good food and resources that the bees are getting ready to swarm and produce a new queen. Whereas on the right, you see that queen cells that are in the middle of the brood or the comb um, are referred to as supersedure or emergency queen cells. And these are queen cells that are built when, um, say, you install a new queen in a colony, the bees don't like that queen, they'll start making their own. Or if, um, a beekeeper happens to go in and, maybe the queen gets squished somehow between frames or what have you, then in that queenless state, the bees will start getting into overdrive to make a new queen to continue to support that colony. Um, so getting kind of into the beef of this presentation, talking about bee breeding and creating your own program for breeding and queen rearing. So when you're thinking of producing queens, you want to first make some assessments and selections. Uh, you need to have enough resources to make strong nucleus colonies or new colonies from what you have. They need to have lots of bees and food to sustain and strengthen these new colonies, as well as have enough resources left behind in the mother colonies, the old colonies, to keep them going as well. Um, Another thing, you want to start early in your queen production so that queens have enough time to mature and then mate and then start building up her own colony, her own workers. And even if you are choosing to do natural mating or instrumental insemination for your uh, queen production, you want to make sure that you're starting early. Um, and a good indicator of when is a good time to start producing queens is when you are seeing drones emerging from the comb. So you see young drones within the colony, not so much flying. Also when there's good weather. So queens will fly for mating flights when it's about 69 to 70 degrees Fahrenheit outside. So once it starts approaching that is when you want to start grafting or making nukes to produce queens. But even with all that, you want to make sure that you have a good, strong population of genetically diverse colonies within your selection, your breeding program. And you want to make sure that you are creating your own selection index of desired traits, and we'll go over some good traits to look for. And then us as beekeepers need to make sure that we are controlling for pests and pathogens. So treating when it's appropriate, getting ahead of the curve so that we're not seeing high levels of Varroa before it's actually, too late to start treating. You want to consider methods for controlled mating, either with artificial insemination or having drone mother colonies situated next to mating apiaries. But really, you want to keep it simple so you're not having to keep track of a large selection index of your desired traits or all of your treatments, and making sure that you are selecting from a good group of colonies for your breeding program. So even if you are trying to rear queens to create a breeding program, the unit of selection from your own apiaries is going to be a full colony, not just the individual workers or queens within a colony. So each worker will have their own strengths and weaknesses. It's just about the frequency of those traits in a whole colony that determines how you want to select your hives to propagate or breed from. So some good traits to look out for a colony is a colony with high performance and one that has low pest and disease levels. So a good indicator of a high performance colony, they are able to fight off diseases, they have good hygienic behavior, they produce a lot of honey. You might want to look out for bees that are with gentle temperament. They'll be a lot easier to work with than more defensive bees. And of course, here in the Pacific Northwest, you want bees that are going to overwinter well. And to determine those colonies with the low pest and disease levels, you want to have a nice frame full of brood that with good brood viability, and again has the reduced susceptibility to pests and pathogens. So you're not going to see a lot of diseases like chalk brood or European foul brood, or have a lot of Varroa that you're seeing on your frames when you're checking out your bees. So besides selecting colonies to breed from, you want to kind of control your queen selection from those colonies. Um, and so right now I'll go over some methods for producing queens, including grafting, which is what I'm more familiar with, as well as a quick overview and summary of some other graft-free queen rearing methods. So The first step in grafting is making a cell builder. So you want a box. Typically, we use just a 10 frame box that is overflowing with nurse bees. And then you want to leave an empty space somewhere in the middle of that box so that you can put your graft frame within that that cell builder. You want to make sure that it's queenless at least a day or two before you put your grafted queens into that colony so that those nurse bees are more likely to start drawing out comb from those queen cells. And you want to load it up with lots of good food, so honey, some pollen frames to surround your graft frame so that it's easier for the nurse bees to feed the developing queen larvae. Then you also want to put in a few brood frames so that as those nurse bees that you put in initially age, that you've got a source of young bees to continue to be feeding those developing queens, even after you've initially put up that cell builder. So after you've left that cell builder queenless for a couple days, before you go in and graft, and before you put your graft in, you want to make sure that you're checking for any emergency queen cells, and you want to cut those out before you put your new graft in so that the bees in there are more likely to accept the queens that you are selecting than the queens that they are trying to rear themselves. So you don't want those nasty emergency queen cells in your cell builder. So after you have set up your cell builder and selected the colonies with the desirable traits that you want to proliferate, you can graft from those colonies. So you want to select a good frame with open brood. And good frames will typically have eggs along the outside of the brood with different stages of larval development kind of going in towards the middle. And a good rule of thumb is that if you see larvae flanked by eggs on any side, that's a good indication that that larva is the right age that you want to be pulling from to graft. So I've highlighted here a couple of larvae. You see they're surrounded by eggs on either side, and these ones are maybe 2 days old. These ones would be okay to graft from. But really you want those larvae like as small as you can get. These larvae up here, down here, those are going to be too big. They're going to be too old to produce good, strong, well-developed queens. If workers do happen to draw out cells with larvae like that, those queens just aren't going to be as well developed as the larvae that are younger and are being fed the royal jelly earlier. So, and likely larvae like these, the larger larvae that are being put in, those queen cells might not even, the workers might just get rid of those before they even draw out comb for those queen cells. So once you've picked out your frame from the colonies you want to graft from, you can graft. So this takes some specialized tools. You need an empty frame with slots to put in a bar like this down here that will have these specialized queen cells that you will scoop larvae out of wax cells from your frame and deposit them into the specialized queen cells. And this can be done with special tools that you can purchase from different beekeeping suppliers. Or you can just take a small piece of wire and like hammer it so that it's flat, so you can just easily scoop the larvae and put them into these specialized queen cells. And the image on the left is just a reminder that you want to be grafting 1 to 2 day old larvae. So the larva that has just hatched and are still young enough to develop into healthy queens. So once you've grafted into your queen cells, you will put your grafted cells into the cell builder and wait for the nurse bees to draw out the comb. And hopefully you will have something that looks like this after you've grafted. So cells with lots of royal jelly, the larvae are just floating on top. It's nice and full. Those queen cells of royal jelly. And you got these nice long pulled down comb of drawn out queen cells. And so that's what you really want to look for when you're grafting for queens. And then once you've done your graft, your cell builder and the nurse bees from your cell builder has drawn out those queen cells. They are about ready to either emerge. You want to have some nukes set up to install those queens, either as queen cells or as emerged queens. So again, you want to have a nuke that's full of nurse bees, lots of good resources, and a brood frame, at least a brood frame, so that there will be more nurse bees to help feed the queen as she's being installed or emerging. And again, you want to set these up a few days before you're ready to install your queen cells or your queens so that it's left in the emergency queenless state. And you want to make sure that you're cutting out your queen cells before installing queens so that those bees are ready to accept a queen and not make their own. So you can either install a queen cell directly into the comb right here, or you can let your queens emerge in cages in the cell builder and then install them into the nucleus colony once they've emerged in these queen cages. All right, so moving on to a quick overview of some different graft-free queen rearing methods. The first one I'll talk about here is the swarm cell method. So in this case, you might have had a good, strong colony that has overwintered. It's got lots of bees, lots of good resources, and in this case, maybe the old queen has already left the colony with half of that colony, and this hive is producing their own queen cells. So in this case, if you go through a colony and you see that they are already creating these swarm cells, there's no queen, if there's a reduced population, you can take these swarm cells, leave 1 or 2 in the original colony, cut out these other cells, and you can either set up nucleus colonies beforehand to put these queen cells into, just like this. You can just pop them in, take some toothpicks, cut out the wax, and just stick them right in to a queenless nucleus colony so that those queens are accepting of that newly emerged queen. Or you let these queens emerge and set up this new colony with a new queen. But if you do decide that you want to take several of those queen cells and put them in different nukes, then by all means, go ahead, stick them in there. And then you want to just kind of close up that nuke for about 3 weeks, let that queen emerge and go on her mating flight, get oriented, go on her mating flight, come back and start laying and producing her own brood. And so you want to wait a few weeks before actually checking on these, uh, the result of this queen cell installation, so that you let the queen have time to settle into these new nucleus colonies. All right, the next method, the nucleus method. Nucleus colonies are great for a lot of things, especially queen rearing. So with this method, you can just take a frame of eggs or very young larvae from a nice, strong colony that you've selected that maybe you want to breed from. You just take that open brood frame, put it into a queenless nucleus colony. Again, the queenless state puts bees in a state of emergency. Like they want to rear a queen as soon as possible so that she can start laying eggs and producing brood as soon as possible to keep that hive going. So if you put just a frame of open brood into a new queenless nucleus colony, those bees will be more likely to take larvae from this new frame of open brood to start making queens from. And again, you want to make sure that with these new nucleus colonies that you're trying to rear these queens from, you are feeding them so that they're staying healthy, but you want to leave them kind of undisturbed so that the queens have time to settle once they've emerged, mated, and are hopefully laying. So this Miller method is a little bit more complicated, a few more steps. Basically, from one of your strong colonies that you've selected to breed from, you want to take this specialized frame that has this jagged edge within the frame that the bees will build from and draw queen cells down from. So you'll put this into the colony that you want to breed from with the queen. You'll let the queen lay eggs in this frame that you've got. And then at one point, you're going to want to remove that queen, let the workers draw out those queen cells. And then you can take queen cells from this specialized frame and put it into new nucleus colonies or requeen old, weak colonies, what have you. And then that's just another example of a way to rear queens. This one is a little more time intensive, and you want to pay attention to the different times, as well as removing a queen from a nice strong colony that you want to breed from. You want to make sure you're saving that queen. So having, again, another nucleus colony to keep her safe in until you are done raising queens from a queenless colony, you can put her back in. And then this other Hopkins method, it takes this specialized frame that is laid on top of frames in a colony so that the workers will draw cells down like you see in the photo. And then you can just cut those cells out and put them into new nucleus colonies or requeen weaker colonies. But in any instance, when you are using these kind of graft-free queen rearing methods, you're taking already produced cells from hives. It's always a good idea to put multiple cells into each colony that you are requeening, or each nucleus colony, so that you have a better chance of a queen that is healthy, well-developed, emerging, and settling into that new colony. So despite what method you want to use for rearing queens, timing is always important. So we kind of went over the different life stages. Um, the the time that you really want to be paying attention to is once the queen cells are capped as larvae, and then after they're capped, they have to be the pupa within those capped cells have to be mature enough to be distributed to the different nucleus or weaker colonies. So around day 13 or 14 is when the queen pupa is fully formed, fully developed. So she won't take as much damage if you decide to remove that queen cell either from a graft frame or a regular frame from the wax. Even if she's jostled around a bit, it won't hurt her as much as younger pupa or queens in cells that aren't capped. So day 13 to 14 is the best time to distribute any queen cells that you want to put into nucleus colonies. And then it goes on even further. Day 16 is when the queen will emerge, and then she'll go on her mating flights a few days later. And then even a few days later is when she will start laying her eggs. All right. So getting into a bit more of the nitty gritty, what's actually inside those long, beautiful abdomen of queens that we see in our colonies. You have these large pear-shaped ovaries that are situated closer to the front, more towards the thorax of reproductively mature queens. And each ovary is made up of strings of ovarials where the ovules are produced, and these will eventually make their way from the tip of the ovary here down towards the different ducts where they develop into fully formed eggs. And these mature eggs will then pass through the oviducts. Here, these structures that lead to this common oviduct. So the eggs will pass through those oviducts and past the spermatheca, which is the organ where the queen will store sperm for her lifetime. And as the eggs pass the spermathecal duct, the queen can release some sperm that will fertilize the eggs before they pass through the this common oviduct or the bursa copulatrix through the stinger where she will deposit eggs into cells. So like I said before, sometimes the queen will lay unfertilized eggs that develop into drones, in which case the queen will not release sperm from the spermatheca. The eggs will just pass straight through to be laid in drone cells. And here the queen has a smooth stinger compared to workers that have the barbed stingers, because this is a specialized stinger that is only really used for laying eggs. However, if you've ever handled a queen and she's not happy, she can still sting you and deliver venom from her venom sac through that stinger to give you a nice little sting. So for natural mating, queens will leave her colony, the hive where she is living, situated, and fly to these drone congregation areas where drones will also leave their hive to fly, where they will be gathered in this area different heights off the ground, just flying around, just waiting for a queen to arrive. And they've got these nice big eyes on the top of their head, all the way out down the sides of their head, so that they can see a queen when she enters the congregation area, and they can start chasing her to try to mate. And this mating occurs in seconds, and for drones, it's kind of a one and done. When a drone sees a queen, he will fly towards her, latch on, and then he will deposit his semen into that bursa copulatrix, that kind of common oviduct within the queen reproductive system. And then once it's all said and done, he will fall off and die. That's it for him. But some strategies that these males can use to make sure that the queen actually keeps the sperm that he's giving her is to leave a mating plug that is made up of some of his endophallus that is being inserted into the queen, and then other seminal fluid products, like a mucus that will just kind of get lodged in the queen's reproductive tract, so that it makes it hard for other drones to come in and mate with her. Because a queen will mate with several drones on a single mating flight, and up to about 20 drones is what she'll mate with on average, or 15 to 20 drones on average is what she'll mate with, to get the sperm that she will keep in that spermatheca for her lifetime. So the queen may go on one or two mating flights, but she only goes on those flights very early after emergence, and only does it once. She won't mate again after that. So if you are going to have this breeding program where you utilize natural mating, you want to have separate drone yards, and a good rule is to have 2 drone hives per queen that you're producing, but that can be kind of a lot. So even if you do have a drone yard and you just have 1 drone colony per queen, that will be good. The idea behind this is to just saturate the area where the queen will be mating with drones that you have selected from colonies with good, strong traits that you want to keep in your breeding program. So you want to saturate the area with those drones, so that there's a better chance that the queens that you've produced will mate with the drones that you've selected. So once you start getting ready to produce queens is when you want to start feeding any drone hives that you have a 1-to-1 sugar syrup, so that it gives the hive the resources it needs to produce strong, healthy drones, as well as feeding the drones what they need to have that energy to then fly out to the drone congregation areas and mate with the queens. And you want to place these drone yards a good distance away, so that there's less chance that the queen is mating with a very genetically similar drone. So here is a quick video of the natural mating. So you see the drone latching on to the queen. This is all happening as they're flying around. He'll insert his sperm into the queen. You see he's kind of falling off. And then he'll leave behind the mating plug. And that's it. That's it for him. So again, one and done. So again, going back to this importance of genetic diversity, one, for mating, this is the reason why queens and drones mate outside of their colony. This reduces the chance of inbreeding, so that there are fewer chances that the progeny from these matings will have similar sex alleles, because that causes issues with diploid drones that will make this funky brood pattern and just create a weaker hive, a weaker colony overall. Um, so the queens and drones mate outside of the colony because if they were to mate within the colony, the drones in the same colony as that queen are either the queen's brothers from a previous queen or her own sons. So you don't want that. You want to reduce that chance of inbreeding. Then, of course, having greater genetic diversity will give you more traits to select from, um, stronger colonies that are less susceptible to pest and disease, have good hygienic behavior, resource allocation. So bees that are better at collecting nectar and bees that are better collecting water, and even other bees that are better collecting propolis. So you want to have all of those traits within your population, so that you can have strong colonies that will overwinter well. And the you also want to be selecting probably from colonies that have been raised in the area where you're trying to create your breeding program, so that they can have this local adaptation to the environment. So, for example, some bees within a certain valley, say in France, if you take those bees, put them in a valley somewhere in Italy, even if the conditions are mostly the same, they might not perform as well as in the original valley that they've been in. So that's that local adaptation to the environment. And just to touch on my own research, as a way of trying to explain the evolution of this local adaptation, um, I am looking at spermathecal content analysis and population genetics to see if sperm from drones that are uh, from the same strain or have these same like local adaptations, um, will perform better than sperm from drones that are uh, less locally adapted to the environment as the queen. So that's just a little bit about my research. I just see that we're kind of coming up on time in the next 15-ish minutes. Um, so that kind of concludes my presentation. Please let me know if you have any questions or if there's any information that you would have liked to see in this presentation that I didn't touch on. Thank you.
Um, Kiersten, we did have one question. Somebody wanted to know, what is a drone mother colony?
Okay, so a drone mother colony is really it's just a separate colony that is like very strong. It produces a good amount of drones, and it has those traits that you are selecting. So really, it's just a colony that is kept separate from your maybe your mating yard, so that the drones from those colonies will be more likely to mate with the queens that you're producing. Um, you could also just have maybe queens that are at the end of her life that are really just drone layers at that point, that maybe had good qualities when she was stronger, maybe younger, that you still want to use those genetics. So that would also be a good option to use as drone colonies.
Have you ever had the luck to see a drone congregation area?
I have not personally, no. Mm-mm.
And I have one more question, which is of the methods that you talked about that don't require grafting. How
Mhm.
many have you tried?
Um, I have tried really just the nucleus method. Um, it's, I guess, essentially like creating a split from a colony. So that's probably the closest that I would have come to using that method for my own um, queen rearing purposes.
There was a follow-up question regarding the drone mother colony.
Uh,
Are you using more drone frames in one of those colonies?
So it would be ideal to use drone frames in those colonies, whether it's ones that you've purchased or the wax frames, or if you take them from old colonies that have nice, good drone brood frames. Um, but I have found that, like, for instance, using these green frames, it's a lot harder for to get the bees to draw wax out onto those. So um
Hmm.
that makes it a little difficult to have like true drone um colonies, but that would be the ideal situation, is to have the bees use the drone frames.
Doesn't that present some problems in terms of Varroa?
Yes, that is a good point. It would um tend to have a lot more varroa in those colonies because of, those drone cells. The varroa like to hide in there. Um, so it's just something that you really have to stay on top of. So,
Yeah.
um, yeah.
Okay, it looks like the person that had questions about the drone mother colony has gotten them answered. Um, are there any other questions for Kiersten? Well, it doesn't look like anyone has any more questions. I want to thank you so much for your presentation tonight, and I really apologize to you and everyone else for the technical difficulties that we had at the beginning of this session. For those of you who are online, if you want to repeat this, this is been recorded and it'll show up on Facebook Live on Ask a Washington Beekeeper, or you can check out WASPA's YouTube channel. It'll be posted there in the next few days. Um, with that, I'm going to say goodbye and thank you again. And we will see everyone next month on the third Thursday at 6:30 for Ask a Washington Beekeeper. Our next presentation has to do with beekeeping equipment. So thank you very much, um, and good night.
Thank you for having me. Have a good night.
Thank you.